All right, so this is the second half of your lecture for chapter 6. Update later, sorry. Okay, so today we're going to talk about the blood, the composition of your blood, what it's made of, how it clots, and then also the lymphatic system. The reason the lymphatic system is put in this chapter is because the lymphatic system is also kind of a circulatory system on its own. It functions very similarly to your circulatory system. How it moves the lymph fluid through that system is similar to how blood moves through your circulatory system via your skeletal muscles. So we'll talk about that at the very end of this chapter, and your book really doesn't go into much depth on your lymphatic system, so that shouldn't be too much of an issue. Okay, but anyways, the blood. So this is where we kind of start to get into the fun part of the class, because blood is what we will be dealing with as phlebotomists. We will be drawing blood. So it's very important to know the composition of the blood and like what you're dealing with, basically. So your blood composition includes plasma, which is 90% water. It includes gases like oxygen, carbon dioxide, and nitrogen. It includes minerals, potassium, calcium, magnesium, sodium. So those are all included in your plasma. It also contains carbs and lipids, proteins, waste products, vitamins, hormones, and drugs. So there is a difference. We talked about it briefly, I know, in a previous lecture. I think I just mentioned it, though, so if you don't remember, don't feel bad. But we talked about how there's plasma and there's serum, and there's a distinct difference between the two. And we will talk more in depth about that in a couple slides here. Okay. So there is also, so aside from just plasma, I'm going to make this bigger, I'm sorry. There we go. That looks better, doesn't it? Sorry about that. Okay, so aside from the plasma in your blood, so the plasma is the fluid portion of your blood. So inside of that fluid matrix of your blood, there are some cellular components, and we call those cellular components the formed elements. So there's three different main types of cellular elements. The first one being your erythrocytes, which are also called your red blood cells. These are the most numerous cells in your body. They are anuclear, which means they don't have a nucleus. They're biconcave, which I wish I could show you like I would draw it out for you guys, but since I can't, I'm thinking. Okay, so there's a picture on page 166 of your red blood cells. You can see how they kind of cave in on both sides. I don't even know how to explain like what that looks like. It's almost like a donut, but the middle part is not a hole. It's like solid. So anyways, they're biconcave and they're intravascular, which means they only perform their duties within the bloodstream. They can't perform any duties outside of the bloodstream. So what is their function? Their function is to carry oxygen and carbon dioxide. So the whole purpose of your circulatory system is to get the oxygen to the tissues of your body, right? Oxygen from your lungs to the tissues of your body. Well, how does it do that? That oxygen actually attaches to your erythrocytes and is carried by your erythrocytes to your tissues. And then once in the capillaries, your red blood cells will diffuse those oxygen molecules through the capillary walls to the tissues, and it will actually pick up the carbon dioxide molecules, the waste products of your tissues, and carry those back to the lungs where they're reoxygenated. So function of your red blood cells is to carry oxygen and carbon dioxide. We also talked about, and when we were talking about the skeletal system, we talked about how your erythrocytes are produced in your bone marrow. So that's definitely an important thing to remember, is that red blood cells are produced in bone marrow. And I'm going to tell you guys in the next slide pretty cool, pretty scary but cool story about this fact about how your erythrocytes are produced in your bone marrow. Okay, so the second type of formed element is your leukocytes. These are also called your white blood cells. So I know a lot of people know that if you have an elevated white blood cell count, it must mean you're sick, right? Well, why is that? Because the function of your leukocytes is to destroy pathogens They're like your little fighter cells in your blood. They're designed to neutralize or destroy harmful agents in your body. So if you have like a autoimmune disorder, your leukocytes actually don't recognize the correct things as pathogenic. They might recognize a part of your blood or tissue that you need as pathogenic and destroy essential parts of your blood or tissues, if that makes sense. So your white blood cells basically attack your own body rather than attacking the pathogens in like a autoimmune disorder. Okay, so these are also formed in your bone marrow, and they're also formed in your lymphatic tissue. So when we talk about the lymphatic system, we'll talk more about how your leukocytes are formed. And there's two different types. So this can get kind of confusing once we get into the two different types of leukocytes. But on page 167, there's a really good chart that shows you all the different types of leukocytes, because there's two types of leukocytes. There's granulocytes and there's agranulocytes. Your granulocytes have three different types. Okay, so try to break it down. Leukocytes, there's two types, granulocytes and agranulocytes. Granulocytes include neutrophils, eosinophils, and basophils. So there's three different cell types of granulocytes. And there's two different cell types of agranulocytes, including monocytes and lymphocytes. So I know it's kind of confusing. I'm not going to ask you, okay, what kind of white blood cell is this? But I do want you guys to recognize that it's kind of like a chain going down. It starts with your leukocytes, and then your granulocytes and agranulocytes are next, and then it goes down from there. Okay? So there's many different types of white blood cells. All of these have different functions in what they target and what they destroy. That's why there's so many different types of autoimmune disorders, because they can have certain leukocytes that are attacking certain things. So, for example, we'll just say, so your basophils, they release histamine and heparin, which enhance the inflammatory response. So if you've got like an overproduction of basophils, or they're recognizing something as pathogenic that's not pathogenic, you might get very edematous, which means you're very swollen. So that's just one example. But we're not going to go into all the autoimmune disorders because that's way above us, but just so you guys are aware. Okay, so you've got your erythrocytes, your leukocytes, and now you've got your thrombocytes. So thrombocytes are a big word for your platelets, and your platelets are an essential part of the coagulation or clotting process. They are actually just cell fragments. They're not a full cell, they're fragments of a cell that are released by what's called a megakaryocyte in your bone marrow. They are the smallest of all the formed elements, and they are the first cells on the scene of like an injury. So when we puncture somebody's vein and we put a little hole in their vein, the first cells that are going to come to that site to initiate clotting and prevent the leakage of blood out of that hole are the thrombocytes or platelets. Okay. I also want to mention that the average adult has about 5 liters of blood. 55% of that is plasma, and 45% of that is the formed elements. So 55% is the liquid portion of your blood, or the fluid portion of your blood, and then 45% are the cells, like your erythrocytes, leukocytes, and thrombocytes. So when you draw a chemistry specimen that you're going to ultimately spin down for the plasma or the fluid part, about half of that will be plasma, and then half of that will be the formed elements that get spun down. Okay, so blood typing. I'm going to show you guys a really good video on this, because usually I'm in class to explain it, and it's very difficult to. I'm a visual person, so I like to see things when they're explained. Just lecturing and talking a bunch of words doesn't really help me much. And with blood type, I think it's a lot easier to understand if you can actually visually see it. So I'm going to show you guys a video that I think explains it really well. But I'll kind of go over it briefly beforehand. So your blood type is inherited. It's determined by the presence or absence of antigens on the surface of your red blood cells. So those erythrocytes that we just talked about, they're antigens. That's really all I can explain them as. And they're actually fixed on the surface of your red blood cells, okay? So your blood either has the ability to develop antibodies directed at the opposite blood type or it doesn't. So basically what I want to say on this slide is if a patient that needs a blood transfusion is given the wrong blood type, their body, if they have those antigens directed at the opposite blood type or the incorrect blood type, will actually attack and destroy or lyse, rupture all of its red blood cells and essentially would kill the patient. So that's why it is so important that you label your specimens and you make sure that the correct label is on the correct tube of blood. Because if you draw a patient and you mislabel a specimen that is then blood typed for a transfusion and that patient gets a transfusion of the wrong blood type, you essentially just killed that patient. Like you yourself, by mislabeling, caused that patient to get the incorrect blood type, which could kill them. So it's super important that you are labeling and you are making sure that you're doing everything step by step correctly. I do have one. This was the example I was talking about, the scary but cool example that I was talking about. So I drew a patient in the ER for type and screen, which is basically like blood typing, for a transfusion. The patient needed a transfusion. And we keep record if they've ever had a transfusion or blood type done. They keep that in record on the patient's medical file, the medical record. And he had many transfusions in the past, this patient. And I drew him for a type and screen. I took it to blood bank, which is where we take our pink top tubes for blood typing. And they ran it, and one of the techs came over to me and said, are you sure that you drew the right patient? And I was like, yeah, like definitely 100%. Like I asked the patient's name and birthday. He told me himself. Nobody else told me. He himself told me. And I didn't have any other labels with me. I didn't have, like, I was 100% sure that I drew that patient. But for some reason, the blood type was completely different from the last time he had a type and screen done. How could that be? How is that possible? Does anybody know? Anybody have any ideas? So, we talked about how your erythrocytes, which determine your blood type, are made in your bone marrow, right? So when a patient has a bone marrow transplant, it's a really confusing process, but basically they kill all of the patient's bone marrow and put the new bone marrow in their bones, which changes or can change their blood type. So I looked in this patient's chart, which you're not supposed to look in the patient's chart unless it has to do with whatever you're aiding in. So you're doing blood work on them. You can look at their blood test or their lab tests. This was in relation to a blood test. So I looked in his chart and I saw that he had recently had a bone marrow transplant. So guess what? His blood type changed. So it was really scary. It makes you second guess yourself for sure, but I want you guys to be aware of that in case it ever happens to you. Don't freak out. Just look in there, see if maybe they had a bone marrow transplant. It's kind of crazy to think about it, but I thought that was a cool story that you guys might enjoy. So, moving on. Okay, so there's two different blood group systems. There's your ABO, so that's if you have blood type A, blood type B, blood type O. This is what most people know if they're A, B, or O. So there's two different antigens. There's the A and then there's the B antigen. So if you have the A blood type, those A antigens are on the surface of your erythrocytes. If you have a B blood type, those B antigens are on the surface of your red blood cells. If you have an O blood type, you don't have antigens on the surface of your red blood cells. Okay? So that makes 4 different blood types, AB, and O. So if you have both A and B antigens on the surface of your erythrocytes, you have blood type AB. I know it seems like really confusing, but after the video, I think it'll make a lot more sense to you guys. And then your RH blood group system, not a lot of people, I think, know if they're RH positive or RH negative. So RH is just an additional antigen, and if you have it, you're positive, and if you don't have it, you're negative. Okay, so you can have AB positive blood, where you have all 3 antigens, the A antigen, the B antigen, and they call it the D antigen for the RH factor. I always call it the RH factor, but whatever works for you, works for you. So I myself have O positive, so I have neither the A or B antigen, but I do have the D antigen, or the RH factor. I think I explained that. So the hemolytic disease of the newborn, I'm not going to go into that. It's kind of confusing, so don't worry about that. But basically, this would be like if the mother is RH positive and the child is RH negative, or vice versa. The mother's blood or the child's blood can attack and kill either the mom or the fetus or both. So the RhoGAM, and it happens only in the second pregnancy for reasons we're not going to get into, but just know that RhoGAM is what they give a pregnant woman if they have this potential issue. Okay, and then so once we do a blood type on a patient, so if a patient needs a blood transfusion, they're first going to do a blood type. They're going to figure out the patient's blood type, and then they're going to do what's called a compatibility test or cross match. So this is where they take the donor's blood, so from the transfusion bag, as well as the patient's blood. They mix it together to determine if they are compatible or suited to be mixed together, because there are some rare occasions where even though they're the same blood type, either the patient's blood interacts negatively with the donor's blood, or the donor's blood might attack the patient's blood, even though it is the same blood type. So you want to make sure that the blood is actually adequate to mix together before giving the transfusion. Okay, so transfusions they can be pretty scary. They have to be done with utter care and focus to make sure that every step along the process is completed accurately. Okay, so I am going to show you now if I can get out of here. How do I get out of here? Okay, I'm going to show you the video now. I also posted this video, the full video, because I'm just going to show you the blood typing portion of it, but the whole first part of the video is a really great video just to review what we're talking about with the elements, the formed elements, and the fluid part of your blood. It's really good if you're a visual learner like I am, so I posted that if you guys want to watch it to review.
Blood drive. Another thing you're going to need to know before you give blood is what type you have. Do you have A, B, AB, or O? These different types all do the job equally well. They just sort of have a different flavor related to your immune system. All the cells in your body have a plasma membrane with specialized glycoprotein markers on them that act like name tags or labels, sort of like, this cell is property of Hank. These markers are your antigens, and your body's immune system is totally fine with your particular antigens, but if it detects antigens from someone else's cells, including viruses or bacteria, then it'll send out antibodies to bind to those markers, often to tag them for destruction by the immune system. Your red blood cells have specialized antigens on them called agglutinogens that activate antibodies that work by binding invading cells to each other, which causes coagulation, or the clumping of blood. Which agglutinogens you have on your erythrocytes defines your blood type, but they're classified in 2 different ways. In the most important blood classification, the kind people are most familiar with, there are only 2 kinds of agglutinogens, simply A and B. B, and your blood can either have one or both or neither of these molecules. So the name of your blood type refers to what kind of agglutinogens you have or don't have. A type has A antigens, B type has B, AB has both, and O has neither. So why do you need to know what type you are before you give or receive blood? Well, like I mentioned, if you have either of these antigens, your body will be fine with it because it doesn't produce any antibodies that label it for attack. So if you don't have a particular antigen on your blood cells, say the type B, then you do have antibodies that are going to label those B antigens for attack should they enter your space. So AB type folks are called universal recipients because they have both antigens and therefore no antibodies for either. So they can accept A or B or AB or O blood. Meanwhile, O type doesn't have A or B antigens, so those folks have antibodies for both. That means that they can only accept other O blood, and yet that lack of antigens means that type O blood can mix with other types of blood without getting attacked, which is why it's known as the universal donor. But just to complicate things a little bit more, you've got a whole other set of antigens with totally different protocol. These are your rhesus or Rh antigens, named after the species of monkey they were first identified in. Much like A and B, you either have the Rh antigens, in which case you're Rh positive, or you don't and are Rh negative. Most of the population is Rh positive, so they don't have the anti-Rh antibodies, which means they can accept either positive or negative blood, but negative types should stick to just the Rh negative blood. And since the presence of AB antigens is controlled by different genes than the Rh ones, we end up with 8 different blood types, 4 separate groups, each with 2 variations. And now, hopefully, you understand why it's so hard to replace blood, and why true blood is not true. I've not actually ever seen that show. Along the way, you also learn the basic components of blood, including erythrocytes, leukocytes, platelets, and plasma.
Okay. So let's get back to our slideshow here. Okay, so now we are going to talk about different blood specimens. Okay, so we're going to get into the difference between serum and plasma now, which is important. The difference between serum and plasma. The difference between serum and plasma will definitely be on your exam, okay? So, I'm going to start right off with that, the difference. Fibrinogen is a protein in your blood. When it is combined with an enzyme called thrombin, it produces what's called fibrin. So fibrin is another protein, and this protein forms a mesh that impedes the flow of blood, so it clots it, okay? So, all of the proteins that I just talked about are enzymes. Fibrinogen, thrombin, and fibrin are all part of the coagulation process of the blood. Serum is the fluid portion of your blood remaining after clotting occurs. So, with that being said, since fibrinogen is used up in the clotting process, forming fibrin, there's no fibrinogen present in serum. So serum clots. Because it clots, all of its fibrinogen is used up in that clotting process. Plasma does not clot, so it still has fibrinogen present. Okay? So the difference between serum and plasma is plasma contains fibrinogen and serum does not. Okay? Pretty simple. Okay, so can you tell the difference between serum and plasma by looking at it? No. They look pretty much the same. So, in order to tell the difference, we have our SST or serum separator tubes, which has what in it? Does anybody remember? It has a silica or clot activator so that it induces the clotting process. It makes the blood clot, okay? And then that clot can be separated in a centrifuge, which spins the blood really, really, really fast. So all of the cellular components go to the bottom, and the serum is left at the top, okay? With plasma, we use an anticoagulant. So usually this is our PST or plasma separator tube, but you're also going to get plasma in any tube that contains an anticoagulant that you spin down, okay? So if you were to spin down a lavender top tube, which we normally don't do because we want to look at the whole blood, but if you were to spin down a lavender top tube, you would have plasma remaining because there's an anticoagulant in it that prevents the clotting process, so the fibrinogen is not used up. I know it sounds really confusing right now, but trust me, it'll get easier, and we'll talk about this over and over and over again, so hopefully it's drilled into your head by the end of this course. But plasma is the fluid portion of the blood separated from the red blood cells, white blood cells, and platelets, not serum, okay? So serum is not the same as plasma. Okay, it contains fibrinogen, and it must be collected in an anticoagulant tube, like I said. So with your PST or plasma separator tubes, we're usually using like a lithium or a sodium heparin as your anticoagulant, which we'll add to our chart next week. And it is used in cases when serum cannot be used. So this is one thing that I would like to mention. Most of the tests that are performed on serum can be done on plasma and vice versa. There are specific tests that can only be done on plasma or can only be done on serum, but most of your basic comprehensive metabolic panel or basic metabolic panel, like all of the general tests can be done on either or. So in a stat situation or an emergency situation, which tube would you use to draw, say, a comprehensive metabolic panel? So if it were me, I would use a PST or a plasma separator tube, because with your serum tubes, you have to wait for that blood to clot before you can spin it down, and that might take up to 30 minutes. So if it can be done on plasma, I'm going to do it on plasma rather than on serum, because it can be spun down right away. I don't have to wait for it to clot, because it's not going to clot. It's got an anticoagulant in the tube. Okay, so in stat situations, you're going to want to use a plasma tube if you can. There are certain tests, like I said, that cannot be done on plasma. So stuff like thyroid studies and a lot of send-out tests can only be done on serum. And then the same is true for some plasma tests. Most of the stuff that can be ran on plasma can be ran on serum, but certain things such as troponins and your pro-BNPs, those have to be done on plasma. Okay, I don't think I want to say anything more about this. Okay. I do want to say your serum and plasma tests are going to mostly be chemistry tests. So your SST and PST tubes we're going to take to the chemistry department of the lab, okay? Moving on. So, aside from serum and plasma, we have whole blood specimens. So these would be your hematology tests. You would take these specimens to the hematology department of the lab, because you're looking at blood as the whole blood. So it's in the same form as it would be in the bloodstream, okay? You do not want it to clot or to separate, so we're going to be using an anticoagulant in it. In our lavender top tubes that we normally take to hematology, it's going to be EDTA. That is the same anticoagulant that is used in our pink top tubes that we take to blood bank. So why do we not want to spin the whole blood specimen down? Okay, so like I said before, if we were to spin the whole blood specimen down, you would end up with plasma rather than whole blood. And in hematology, they're looking at the formed elements within the blood. So we don't want to spin those formed elements out of the blood into a big clot at the bottom. So we're looking at the erythrocytes, the leukocytes, and the thrombocytes, and we're looking at disorders of those components within the blood, or issues within those components in the blood. So a CBC would be an example of a whole blood test, complete blood count, because they're checking out the erythrocytes, white blood cells, and your platelets. Okay, so it must be mixed a minimum of 2 minutes just prior to the test, blah, blah, blah. Common errors with whole blood specimens is that they come clotted. So how could that happen? How could we get a clotted whole blood specimen if there's anticoagulant in the tube? So if you get too much blood in your tube, that could cause the blood to clot, because those tubes have a preset amount of vacuum. So they take in just the right amount of blood for the amount of anticoagulant that they have. So the ratio of blood to anticoagulant is perfect with what they have. We have gotten specimens that are clotted from nurses that take a syringe, and rather than just letting the blood get sucked into the tube via the vacuum system, they squirt the blood into the tube, and they actually squirt more blood into the tube than is needed, or so the ratio is off, and that causes clotting. Also, with microtainers, when we're doing finger sticks, the blood can clot very easily in those if you fill them up too much, because there's not a vacuum on those, since we're dripping the blood from the finger stick into the tube. That's really easy to get too much blood in those tubes and cause clotting. So if it's clotting, they can't test it because those formed elements are not in the right values. Okay, what else do I want to say? Okay, so when I took this class, the one thing I didn't understand was why would we need an anticoagulant in a blood tube if the blood is in the same form as it is in the bloodstream? It doesn't clot in our bloodstream, so why would it clot outside of the bloodstream if we didn't have an anticoagulant tube? The reason for this is when blood is stagnant, it tends to clot. It needs that constant flow, so the heart is constantly pumping it through your circulatory system, which keeps it from getting stagnant and clotting. That's why people with like AFib, they get blood clots or DVTs, because their blood is not flowing adequately because their heart's not pumping adequately. So that's how people get blood clots and stuff like that in their circulatory system. Okay, that's all I want to say about that. Okay, so this picture is on page 170, and this just on my PowerPoint I just have showing the plasma. On page 170 it shows both plasma and serum, and you can see you can't really tell the difference between the two, but this is how it will look once you spin your tube down in a centrifuge. The plasma, where's my mouse? The plasma will be on the top part here, and like I said, this is 55% liquid. In plasma, it has fibrinogen. In serum, it does not, because it's used up in the clotting process. Then we have your buffy coat, which is like your white blood cells and platelets. If we were using an SST tube or a PST tube that had the thixotropic gel, so that, remember that white stuff that I showed you at the bottom? That would be present between your buffy coat and your plasma, and that would just basically, it's like a barrier that keeps the plasma from mixing back in with the formed elements. Okay, so the white blood cells and platelets are in your buffy coat, and then all of your red blood cells are going to be at the bottom, and this is because your red blood cells are the most dense. Your red blood cells are the most dense, so when you spin them down, the more dense material is going to go to the bottom. And then your white blood cells and platelets, they're smaller than your red blood cells and in less concentrate, like there's not as much, so they're going to be the next, and then your plasma is going to be the least dense, so it's going to come to the top, okay? So when they're testing your blood, they're actually going to be taking a little sample of the plasma. In a whole blood specimen, it would just all still be mixed. It would look like red, like it just came out of the body, okay? And they would take just a specimen sample from the top of that, okay? Okay. So there are many different disorders of the blood. I do want you guys to know these disorders and some of the tests that are performed on them. So a lot of these tests are done in hematology or in blood bank. So your whole blood specimens are hematology and blood bank. Your chemistry specimens are going to be your SST, PST, or serum and plasma. And then your coagulation specimens are going to be like your light blue top, which will be your clotting factors, which we will talk about coming up here. Okay, so any problems that you have in clotting. Okay, so box 6-4 on page 171, this is where it lists the disorders and your diagnostic tests. So anemia, that's like you have a low number of red blood cells. Leukemia would be like a type of cancer where you have an overproduction or too much. I'm sorry, I'm like tripping over myself here. Leukemia would be where you have a lot of immature blood cells. Okay. Leukocytosis would be increased white blood cells. Polycythemia we talked about would be high number of red blood cells. Remember we talked about how that bloodletting is a therapy for people that have polycythemia. Thrombocytosis is an increased number of platelets, and then thrombocytopenia would be like a low number of platelets, so you might need like a transfusion of platelets. So that's what's cool also about transfusions is you can separate the different elements of the blood. So if you only need platelets, like if you have a low platelet count, you don't need more red blood cells, you don't need more white blood cells, you only need the platelets. We can separate those platelets out from the transfer or from the donor sample and just give you the platelets that you need rather than the whole pint of blood that they donated. Okay, and then diagnostic tests would be ABO and RH type, so that would be like blood typing. We call it a type and screen at our facility. A lot of people call it ABO and RH. Bone marrow, so we won't ever have to collect bone marrow, but the lab techs and doctors will work together to collect a bone marrow specimen they will analyze. Complete blood count, that's a very common test. Looking at a whole blood specimen, like I said, it looks at your erythrocytes, leukocytes, and platelets. A cross match, that's what I talked about when we're mixing the donor blood with the patient's blood to make sure that the transfusion will go correctly and that they won't actually I don't have a word. The word is not coming to my brain, but it just makes sure that the donor blood and the patient's blood will mix together. And then a differential, that's usually part of a complete blood count. Erythrocyte sedimentation rate. This is basically looks at like swelling and inflammation and stuff like that. Hematocrit and hemoglobin. This is part of a complete blood count, and iron. Okay, so hemostasis, this is basically how your body heals like a wound, okay? So when we puncture a vein, this is how your body stops the flow of, or stops the bleeding and heals that little puncture in the vein. So I thought a lot about this, and the process of hemostasis, they go into a lot of depth in your book about it, but I really don't find it that necessary for you to know the whole process of it. So I'm not going to test you on the process of hemostasis itself, but I will be testing you on the tests and disorders associated with the clotting process or the coagulation process. So I'm just going to briefly go over it with you guys. So hemostasis is basically the arrest of bleeding after injury. There are 4 different steps in this process. There's vasoconstriction, which is a reduction in the diameter of your blood vessel. This is caused by contraction of your smooth muscle fibers within the tunica media. We remember talking about the tunica media, which is the middle section of the wall of the vein. So when that blood vessel constricts, it actually prevents it slows the flow of blood to that spot so that less blood will leak out, okay? The second part is the formation of a primary platelet plug. So we kind of talked about this as the adhesion, activation, and aggregation of platelets. So your platelets will clump together at that site, at the site of injury, and kind of form like in combination with fibrin, they form like a mesh or lattice over top of the wound to prevent the blood from leaking out of that hole, okay? And then the third step would be the progression to a blood clot. So there's 2 different steps in this step. There's 2 different stages in this step, I guess I should say. There's amplification. This is where thrombin is added to the blood or the platelets in order to generate the fibrin that I talked about, which is needed to reinforce that platelet plug. And then propagation. This is where there's a big burst of thrombin, and that triggers the activation of certain factors that we're not going to get into depth with, that basically cross-link with the fibrin plug, and that creates that meshwork that I talked about, and that keeps the blood from escaping that that vein or injury site, okay? And then the fourth step would be fibrinolysis, which removes the clots after like the healing process is completed. So once that hole is closed over, the clot kind of dissolves in the stage of fibrinolysis. Okay, so that's really like all I want you guys to know about the hemostatic process. So we're going to skip through this. So what we can see, here's where the injury occurs and your erythrocytes are leaking out of there. So then the first step, your formation of your platelet plug. So these are all little platelets that aggregate right at the site to prevent leakage of your erythrocytes. And then you've got your secondary hemostatic plug formation. So this is where your thrombin is combined with your platelets to form fibrin. And that's that meshwork that I talked about. It really links and kind of glues all of the platelets and red blood cells together so that you don't leak blood. Okay, so we're going to skip through all this. I also posted a video, if you guys want to know more about how your blood clots and coagulates, I posted a video on this hemostatic process that goes more into depth, and it's more visual, so if you want to look at that, you can. Okay, so your examples of disorders and diagnostic tests are on page 177, box 66. So most of your tests that we're going to do for clotting are going to go to the coagulation department, which, imagine that, coagulation is another word for clotting. So a lot of your blood clotting disorders are going to be tested in a light blue top, okay? So our light blue top is our coagulation tube, goes to the coagulation department. And what did we talk about was super important? I don't know if we actually I don't know if we talked about it. With your light blue top, it has to be completely full. Like, you cannot, with all the other tubes, it's acceptable to be a little bit short on a draw. You can't have more blood in the tube than what is required, but you can have less. With a light blue top or coag tube, you have to have exactly the amount that is suggested on the tube, okay? So I will show you that in class, and we will go over that more next week, because next week we talk less about the processes and functions of your body systems and more about the actual testing and blood drawing, okay? So we'll talk about that next week. But these are common tests for coagulation: bleeding time, D-dimer, factor assays, platelet function assay, prothrombin time. This is going to be your D-dimer and these two down here. We refer to these as a PT and a PTT. These three are by far the most common coagulation tests you're going to see. And your PT and your PTT, pretty much everyone that's on a blood thinner will have this done. And when you first get put on a blood thinner, it'll be done weekly to make sure that your blood is not too thin and not too thick. Or not too thick, I guess I should say. A lot of times they have a machine that can do it with a finger stick, but if their finger stick or their values are off, they have to come in and get an actual blood draw, because it's a lot more accurate than the finger stick. So we'll talk again more about the PT and the PTT next week, but know, at least, for the love of God, at least know D-dimer, PT, and PTT. Those will most likely, one of those will most likely be on your exam, okay? All right, already into the lymphatic system. Okay, so the functions of the lymphatic system is to return tissue fluid to your bloodstream. It protects your body by removing microorganisms and impurities, processes lymphocytes, delivers fats absorbed from the small intestine into the bloodstream. So, lymph fluid is very similar to plasma. However, it's 95% water. So it's mostly water. And like I said at the beginning of this video, your lymphatic system is very similar to your circulatory system, okay? It kind of like almost lines it, and it actually links into your circulatory system in your jugular and your subclavian veins in your neck. So I wish I could draw this out for you, but so we talked about in your circulatory system how you've got your arteries, your arterioles are a little bit smaller, then down to your capillaries, which are the smallest, your venules a little bigger, veins are the biggest, right? So there's a similar string in your lymphatic system. Your lymph capillaries are the smallest, and this is where the excess fluid that's found in your tissues is diffused into your lymph capillaries, and then it is carried through the lymphatic system, and it's filtered in your lymphatic system before it's returned to the blood through the subclavian or jugular veins. So it starts in your lymph capillaries. From your lymph capillaries, it moves into bigger lymphatic vessels. Through your lymphatic vessels, it travels to your lymph nodes. In your lymph nodes, this is where it traps and destroys bacteria and foreign matter. This is kind of like the purification process of the fluid. So you don't want to put pathogenic fluid or fluid that's got bacteria in it back into your bloodstream. No. So in the lymph nodes, that's where it filters it and kind of purifies the excess tissue fluid. So from your lymph nodes, it goes to your lymphatic ducts. And you have two main lymphatic ducts. You have your right lymphatic duct and your thoracic duct. And those lymphatic ducts drain into your subclavian and your jugular veins in your neck. Okay, so all that excess tissue fluid that was in your tissues eventually ends back up in your bloodstream. So your body is constantly doing this. Let's go on. Okay, so here you can see where it links up to your right lymphatic duct and your thoracic duct. They link into your subclavian and your jugular veins, okay? And then back into your circulatory system. Oh, I kind of already went over this, but I'll read it again. So lymph is your excess tissue fluid that filters into your lymphatic capillaries. Your capillaries join to form larger vessels that empty into terminal vessels, which are your lymphatic ducts, and then those two ducts empty into the large veins in your upper body. And like I said, the lymph is moved by skeletal muscle contraction, similar to your circulatory system. While you have your heart that's pumping the blood through your circulatory system, you also have that skeletal muscle action that helps the blood move through the veins back to the heart, where there's less of a force behind in the veins. And then your lymph passes through lymph nodes that filter out the impurities. So we talked about that again. So with that being said, I would like to make a point that we never draw we never draw from an arm if a patient has had lymph nodes removed on that side. So if they've had lymph nodes removed in their axillary area on their right arm, we're not drawing from their right arm. Can anybody tell me why? So since your lymphatic system filters out the impurities in your tissue fluid, they have less of a chance of fighting infection if they don't have those lymph nodes. Okay, so if their lymph nodes are removed, that tissue fluid that's being taken through their lymphatic system has less chances for filtering the impurities out. They also cannot get rid of the excess tissue fluid as adequately as most people. So if trauma or injury, a common body response is inflammation. So it sends excess tissue fluid to that area, and that's when they get edemas, where their hand just puffs up. And I'll show you some examples in class when I get back, but their hand will just puff right up. It'll be full of water. Like you can actually push into it and your fingerprint will stay like a hole, like a pit in their skin. So a lot of times you can kind of tell if their arm is a little swollen. You might be able to tell if they've had lymph nodes removed. It's common for women that have mastectomies that they get lymph nodes removed. So good call would be, if they say they've had a mastectomy, not to draw from that side. Even if they don't know if they've had lymph nodes removed, it's just a good safety precaution to not draw from that side. Just go with the other side. And most people will tell you too, like if they've had lymph nodes removed, their doctors advise them that they cannot have blood pressures taken on that side, they cannot have blood draws on that side. Not only that, but if they are in the hospital, there should be a sign above their bed that says no blood pressures or blood draws on the right side, or they might have a sleeve on their arm, like a big red sleeve that says no blood pressures or blood draws on this side, okay? So if they're not aware of it, so if they're, maybe they have dementia or something, they don't remember, there should be a sleeve or a sign above their bed that will tell you that, okay? And then on page 179, box 6-7, these are examples of your lymphatic system disorders and common diagnostic tests. The most common ones we're going to see is a bone marrow biopsy, complete blood count, and a culture and sensitivity. I mean, obviously we get mono tests, but these ones are send outs at our lab, so we send them to a different facility. We have like an Epstein-Barr, which is what causes mono tests that we do at our facility, but for the full mono test, we usually send that out. And so anyway, so know these disorders and watch those videos that I posted. If you guys have any questions, do not hesitate to ask. And I will see you in class on Monday. Super exciting. I will grade your exams on the plane tomorrow. So I'm coming home tomorrow and I will be grading those while I'm on the plane. And I will give you your exam Tuesday, but your next exam is going to be on Monday, okay? So your exam for chapters 5, 6, and 14 from this week is going to be this upcoming Tuesday. The following exam, your third exam, will be on Monday of the next week, okay? Let me pull up my calendar so I can give you guys exact dates on that. So exam 2 will be Tuesday the 10th. And then exam 3 will be Monday the 16th, okay? Alrighty.